Previously I mentioned that the interior of a protein is largely hydrophobic, meaning that there's almost no water to be found there. Water is a hydrogen bond donor and a hydrogen bond acceptor, and it would typically hydrogen bond with amide groups. In the absence of water, what happens is that one amide group on an amino acid residue on one portion of the chain hydrogen bonds with the amino acid of another. The carbonyl is a hydrogen bond acceptor, the NH is a hydrogen bond donor, and those two getting involved in a hydrogen bond. Now, the hydrogen bonds aren't random, but in fact there's patterns of hydrogen bonds that emerge, and these are the patterns of hydrogen bonds that we've come to recognize as secondary structure, like alpha helix and beta sheet. We're going to take a look at those patterns of hydrogen bonding or secondary structures in this particular webcast. And what you see in this cartoon is the picture of the alpha helix. Here we can look at the chain and identify alpha carbons. When we spot an alpha carbon, we can look to the atom on the left. In this case, we see it's an NH, and an atom on the right, it's the carbonyl. And so we note that the chain propagates in the direction N to C along the pathway where the helix spirals upward. Now, if you were to use your right hand and let your fingers curl around that chain, they'll travel in the same direction as your thumb, and so we know that this is a right-handed helix. If you try to do the same thing with your left hand, what's going to happen is your fingers are going to travel along the chain in one direction, and your thumb's going to be pointing in the opposite direction, so we know that this is not the left-handed helix. Now, by adjustments in phi and psi, remember that the amide carbon-nitrogen bond is rigid and planar and fixed, but adjustments of phi and psi allow us to position the carbonyl on one residue adjacent to or directly below the NH of a residue that's 4 residues away. And that allows for the hydrogen bond to set up and fix that secondary structure, the alpha helix, in place. We can see that there are multiple hydrogen bonds, and they're all parallel to one another along the backbone of that polypeptide alpha helix segment. It turns out that there's about 3.6 actually residues in one turn of the alpha helix. Also, a consequence of all of this structure, you can notice that each of the carbonyls are pointing in the same direction. Here they're all pointing up along that helix axis. They lie parallel to the helix axis. And so there's a dipole moment associated with each carbonyl. They all add together in the same way, and so that you can see that overall there's going to be a very large dipole moment that is going to travel parallel to the helix axis, and it's going to run from the N to the C direction. And so we can always remember that there's going to be, because of the additivity of these individual dipoles, a very large dipole moment associated with the alpha helix. I've made some models. You can try to make some models yourself, but what you'll notice is some of the hydrogen bonding interactions along the chain. So the adjacency of the NH and the carbonyl, as you can see here, allow for the hydrogen bond. And now if we spin this around, you can see different orientations of this. You can see different orientations that show that hydrogen bonding, and now you can imagine multiple hydrogen bondings that would help rigidify that structure. We can also look at what's more or less the typical cartoon representation of the alpha helix, and I've now shown you where the side chains are projecting, and so we see multiple side chains coming, basically radiating outward from that alpha helix. And so you can pick up, for example, there's lysine, and there's an arginine, there's a cysteine, there's probably an aspartate or a glutamate, and there's a leucine group. So you can see an alanine and a methionine. There's all kinds of side chains, and they basically radiate outward from the center of that alpha helix axis. In other words, the alpha helix provides this rigid scaffold which projects the side chains orienting outward radially from that structure. The second type of secondary structure or hydrogen bonding pattern that emerges is that of the beta sheet that's shown here. And if we just look at some strands that are lying adjacent to one another, we again pick out the alpha helix. We pick out the alpha carbon. We notice that the nitrogen is on the top, the carbonyl is on the bottom, and so we see that this chain runs from the top to the bottom in the N to C direction. The adjacent chain, there's the alpha carbon, has its nitrogen on the bottom and its carbonyl on the top, and so this chain is running in the opposite direction. Now, I've positioned these chains in opposite directions, and they're basically going to be forming what's the anti-parallel beta sheet. And the hydrogen bond pattern that emerges is between strands of these parallel lying adjacent polypeptide segments. So the strands and the hydrogen bonding pattern that emerges is shown here. You can see the blue lines represent the hydrogen bond donor to acceptor orientation, and that they're lining up, and that as a consequence of all of this, what we end up with is a segment or a region of a sheet-like structure where there is largely planar, although you're going to see that these really in three-dimensional space actually have quite a bit of twist to them, but over a fairly large region they're going to be essentially a planar kind of orientation structure. On the right-hand side, I've placed the strands in slightly different forms. So there's the alpha carbon, and we see that the nitrogen and carbonyl run that direction. And here's another unit where you can see the nitrogen and the carbonyl are also running in the same direction. These would be making up the parallel beta sheets, and their hydrogen bonding pattern is just going to be slightly different. Basically, if you can just make a strand and then try to translate it, in order to maximize hydrogen bonding, this is the best you can do in the parallel sheet, where there's actually not this parallel orientation between the hydrogen bonds, but this slight skew basically between adjacent units. Let's take a closer look at the three-dimensional structure of a beta strand, and what I would encourage you to do is to go ahead and make a model. If you make a model of 3 peptide segments, what you're going to notice is, and try to position all of the groups, the backbone atoms, the carbon alpha, nitrogen, and carbonyl, of those 3 amino acid segments in a plane, you can see that the residues run below and above the plane, alternating from one adjacent residue to the next. And that's really what we have in this, what's known as the beta sheet that's pleated. The parallel and anti-parallel beta pleated sheet have adjacent residues, the side chains, that will be projecting above and below the plane. Here you can see the alpha carbon, its side chain on one residue. If we travel along the adjacent residue, the alpha carbon and its side chain are projected upward. And so there's going to be a puckering of the sheet. Again, it's set by the torsion angles phi and psi that show up in the Ramachandran plot, largely or closely to 180 degrees. But it does so in a way that makes this pleat. There's a because of the tetrahedral alpha carbon geometry, there's this pleated structure with the side chains orienting alternately up and down. Now, in the case, there's a three-dimensional structure of it. You can see that the adjacent strands have the hydrogen bonding between them, and if we look at and stop this movie right on its side, you can see the pleat in the sheet, and now you can see that the amino acids on adjacent units are above and below the plane of that sheet. And finally, I have for you what's known as the beta turn. The beta turn is going to have 3 segments, and so starting with residue at the alpha carbon, which we'll call segment I, then I plus 1, I plus 2, and I plus 3 is here. It's the amino group of the I plus 3 that hydrogen bonds with the carbonyl of the residue I, and there's, by adjusting phi and psi around each of those adjacent segments, it's possible for that beta turn to set up the anti-parallel beta pleated sheet. So we're going to have an N direction, we'll have a C direction, and by making this turn, we're going to be able to get adjacent strands to run anti-parallel to one another. That's known as the beta turn.